The CRISPR technology allows scientists to make changes to the DNA in cells that could allow us to cure genetic disease. You might be interested to know that the CRISPR technology came about through a basic research project that was aimed at discovering how bacteria fight viral infections. Bacteria have to deal with viruses in their environment, and we can think about a viral infection like a ticking time bomb. A bacterium has only a few minutes to defuse the bomb before it gets destroyed. So many bacteria have in their cells an adaptive immune system called CRISPR that allows them to detect viral DNA and destroy it. Part of the CRISPR system is a protein called Cas9 that's able to seek out and cut and eventually degrade viral DNA in a specific way. And it was through our research to understand the activity of this protein Cas9 that we realized that we could harness its function as a genetic engineering technology, a way for scientists to delete or insert specific bits of DNA into cells with incredible precision that would offer opportunities to do things that really haven't been possible in the past. When viruses infect a cell, they inject their DNA. And in a bacterium, the CRISPR system allows that DNA to be plucked out of the virus and inserted in little bits into the chromosome, the DNA of the bacterium. And these integrated bits of viral DNA get inserted at a site called CRISPR. It's a mechanism that allows cells to record over time the viruses that they have been exposed to. And importantly, those bits of DNA are passed on to the cell's progeny. So cells are protected from viruses not only in one generation, but over many generations of cells. This allows the cells to keep a record of infection. Once those bits of DNA have been inserted into the bacterial chromosome, the cell then makes a little copy of a molecule called RNA that is an exact replicate of the viral DNA. RNA is a chemical cousin of DNA, and it allows interaction with DNA molecules that have a matching sequence. So those little bits of RNA from the CRISPR locus they bind to protein called Cas9 and form a complex that functions like a sentinel in the cell. It searches through all of the DNA in the cell to find sites that match the sequences in the bound RNAs. And when those sites are found, this complex associates with that DNA and allows the Cas9 cleaver to cut up the viral DNA. Makes a very precise break. So we can think of the Cas9 RNA sentinel complex like a pair of scissors that can cut DNA. It makes a double-stranded break in the DNA helix. And importantly, this complex is programmable. So it can be programmed to recognize particular DNA sequences and make a break in the DNA at that site. As I'm going to tell you now, we recognized that that activity could be harnessed for genome engineering to allow cells to make a very precise change to the DNA at the site where this break was introduced. That's sort of analogous to the way we use a word processing program to fix a typo in a document. The reason we envisioned using the CRISPR system for genome engineering is because cells have the ability to detect broken DNA and repair it. So when a plant or animal cell detects a double-stranded break in its DNA, it can fix that break either by pasting together the ends of the broken DNA with a little tiny change in the sequence at that position, or it can repair the break by integrating a new piece of DNA at the site of the cut. So if we have a way to introduce double-stranded breaks into DNA at precise places, we can trigger cells to repair those breaks by either the disruption or incorporation of new genetic information. The technology is being used to make very precise changes that allow us to study the way that these changes in the cell's DNA affect either a tissue or, in this case, an entire organism. But we have to also consider that the CRISPR technology can be used for things like enhancement. Imagine that we could try to engineer humans that have enhanced properties, such as stronger bones or less susceptibility to cardiovascular disease, or even to have properties that we would consider maybe to be desirable, like a different eye color or to be taller, things like that. Designer humans, if you will. Genome-engineered humans are not with us yet, but this is no longer science fiction. Genome-engineered animals and plants are happening right now. And this puts in front of all of us a huge responsibility to consider carefully both the unintended consequences as well as the intended impacts of a scientific breakthrough.